Great. Thank you. Thank you for the invitation, and it's a pleasure to be here, and the kind words as well. I want to continue the theme of adaptation. I would state that I probably love adaptation more than Sarah does, but since the inception of agriculture more than 10,000 years ago, I almost said million, 10,000 years ago, crop production has been plagued by abiotic stresses such as drought and temperature extremes and salinization of soils. Those stresses are now increasing both in frequency and severity as a result of climate change. And we only have a few options ahead of us when we deal with how we're going to feed ourselves through the rest of this century. We either find new food sources, and there are people looking into that. Of course, the easy one would be to stem the tide of greenhouse gases into the atmosphere, but we appear to lack the social and political will to do that. The other option is, of course, to generate crops that are abiotic stress tolerant. Now, when we think about that, we can start with a couple of questions about plants and how they respond to stress. And it's very interesting because all plants have the ability to perceive and respond to stress, but very few have the ability to adapt to stress. And people have studied this for a long time, but in essence, this was the premise that led to an intensive scientific undertaking over the last several decades to generate crop-tolerant crops, either by breeding traits from native plants that had clearly adapted to the stresses of interest in nature, or by genetic modifications in one form or another. Now, these strategies were both based on the assumption that plants operate as individuals and that they adapt by what we would call a normal individualistic Darwinian process. And neither of these processes have borne much fruit. Now, one of the reasons for it, we believe, is that, first of all, there's no such thing as an individual in nature, neither in the plant or animal community. All of us in here are just meat bags full of microbes. You know that. Plants are veg bags full of microbes as well. And they've been studied for an awful long time. Now, with plants, it's kind of interesting because all plants are symbiotic with microorganisms. And the plants present three different microhabitats, I guess if you say, or habitats to these microbes. There's the root zone or the rhizosphere and the area surrounding the roots in the soil. There's the phyllosphere, the canopy, and then there's the endosphere, which is inside the plant. And the endosphere is what I'm going to be talking about because I've focused most of our attention on fungal endophytes. Now, these are fungi that live entirely inside the plants. They don't act like mycorrhizae, so they don't grow out into the soil. They reside inside the plant throughout most of their life. Now, this is a particularly interesting group of organisms because their association with plants can span what's known as a symbiotic continuum. And it can vary from parasitism to mutualism. So don't ever think that symbiosis just means beneficial. It can mean a lot of different things. Within this group, there are various phenomena that also define them, one of which is that individual strains of a specific fungus can have the ability to switch symbiotic lifestyles and can move between parasitism and mutualism, either based on the environmental landscape that they see or on the genetic landscape that they see. Let's see, what else? Oh yes, and there are a few different classes, 4 different classes at least, of fungal endophytes that can be differentiated a few different ways. Now, the interest that we garnished over fungal endophytes really had to do with looking at how plants adapt to stress. And this is a little bit old now, but I thought I'd just briefly go through this because it provides a background for everything else. We started looking at plants that grow in these geothermal soils in Yellowstone National Park. These are soils that get up to about 65 C in the summer, and then they'll drop to about 20 C in the winter, but they never freeze. They're also very dry in the summer because it's not raining, and they're very wet in the winter because it's snowing. And so there's this big annual cycle that goes on, but these plants that have been there for millennia have to deal with these high temperatures in the summer. So we were actually doing a big microbial study there. We looked at these plants, and what we found was that they in fact were symbiotic with fungal endophytes in predominantly one endophytic species, Curvularia protuberata, although that's not critical to this. And we became interested in this plant in particular. This is tropical panic grass. There are about 8 to 10 or 12 species of plants that live in these geothermal soils, but tropical panic grass could grow in the hottest soils and grew isolated from the other plants as well. So we looked at a couple hundred isolates from around the park, and what we saw was that they all had the same fungus in them. Now this was atypical because usually when you look at plant communities, there's quite a diversity of fungal endophytes associated with them. But here there was pretty much just one. So it turned really rapidly into a series of Sesame Street type studies. There's only one thing in these plants, gee, I wonder if it's ecologically significant or not. And so we tested that first in the lab, looking at what happens when we develop these geothermal soil simulators where we could heat the root zones to whatever temperatures we want. In these graphs, NS stands for non-symbiotic, S stands for symbiotic. And this is a typical experiment we did at the time where we would heat the root zones, in this case to 70 C during the days. We turned it down to 37 at night. The symbiotic plants survived throughout the 10-day experiment. The non-symbiotic plants died within a day. And to confirm that this, in fact, was significant from an ecological perspective, we went back to Yellowstone. We carried out 1,000 pounds of soil and pasteurized it, brought it back in, put it in bottomless tubs, and then we put in those tubs both symbiotic and non-symbiotic plants that we had generated in the greenhouse. And we just came back a year later and said, what happened? And what we found was that in soils that had 35 C or higher temperatures, if the fungus was not in the plants did not survive. Below 35 C, fitness was greatly enhanced by having the fungus present. Okay, so it was ecological confirmation of a lab study, and it was, I believe, the first time where an endophytic association could be assigned significance outside the realm of just nutrition going from the plant to the fungus. In this case, it was survival of both the species. Now, most importantly from this, and I forgot to mention, is that we could tease this system apart. Right? So we could grow the fungus on artificial media, and we could grow the plants free of the fungus. And azenic growth of either one of those had temperature maxima of 38 C. So neither the fungus nor the plant was heat tolerant. But when we put them back together, they achieved thermotolerance. So it was symbiotic communication that was driving the heat tolerance in this case. When the fungus was removed from the plants, this is probably the most important statement I can make through the whole talk, those plants were not adapted to that habitat. The plants themselves did not adapt to that habitat. It was a symbiosis that adapted the plants. Okay, so there were a lot of questions at the time. One was, is this very specific? This is like a freakish event and very unique to Yellowstone or these species. And so we started asking if the fungus could get into other plants, grow asymptomatically, and if it did, what it did inside those plants. So here's just an example of the type of things we saw. These are watermelon plants grown hydroponically over a temperature gradient. The ones on the left don't have the fungus. The ones on the right do have the fungus. And so you can see that without the fungus, the watermelon plants grow to about 38 C. It's a pretty typical temperature for most plants before they start wilting and dying. But you put the fungus in, and the watermelon plants can grow to 50 C without a problem. Now, so that was cool because we could buy watermelon seeds anytime we wanted to, but the more cool part about this was the fact that we took the fungus out of a grass, out of a monocot, and we put it into a eudicot, and not only did it go through the same developmental process to get into that plant and grow in it, but it communicated the same way. So across these deep lineages of plants, here you have this fungus that appears to have no sense of host range and can communicate the same way. Now, not to complicate the issue, but I just have to throw this in there as well, the fungus also has a double-stranded RNA virus in it, and the virus is required for heat tolerance as well. So it's a tripartite symbiosis, and they've all tagged together in this I don't want to say parasitic way, but they're all codependent. It's extreme codependency. And they're all required for survival of each other. Okay. Well, that was a stimulus to begin looking in other habitats and asking if the same thing happened or not. And so we looked at habitats in various places around the world with different types of stresses. And in every case where we could go in, identify the stressor, and tease the system apart, we found exactly the same thing as we did in Yellowstone, with different fungi and with different plants. But we then magically had this predictive capability of going into these places and saying, hey, guess what? You know the plants over there? They're there because there's a fungus inside of them conferring tolerance to these stresses that are occurring in this environment. And so it appears to be a fairly common phenomenon. Maybe not the only phenomenon out there, but a common one. Okay, so how does it happen? How does stress tolerance happen within these symbiotic associations? Well, what's shown here is what happens is the understanding of how plants adapt to stress through a plant physiological perspective. Plants are hit with a stress, and the first thing that happens is there's a burst of reactive oxygen, and that triggers then a series of events. Eventually, a series of metabolic cascades are activated, all in an attempt to mitigate the impacts of this stress. So we developed some assays. One of them is a reactive oxygen assay to ask what happens in these plants that are symbiotic or not. And the long story short is that the symbiotic plants that are hit with stress do not have a burst of oxygen, of reactive oxygen. The non-symbiotic plants do exactly what they're supposed to. They're hit with stress, there's a burst of reactive oxygen, and then they turn on all these cascades, and then they die under the stress that we give them. But the symbiotic plants don't have the burst of reactive oxygen, and therefore don't turn on these pathways. So they do something very atypical from a plant physiological perspective. So if you have a non-symbiotic plant, you get your ROS, the plants freak out, they turn on everything to try to mitigate impacts, and they end up succumbing to that. The symbiotic plants have to have the right endophyte for the stress. And if they do, there's no reactive oxygen, and they just sort of chill out there and wait for the stress to go away. Now, we've kind of built a working model around this and separated metabolic responses into two types, the general metabolic response, but also the stress response. And I just want to take you through that briefly. The stress response, which is of most equal interest to me, is, as I mentioned, there's not a reactive oxygen burst that goes on. Throughout the entire process, the plants appear to maintain metabolic balance. And that metabolic balance is in part why they don't see such a big reactive oxygen burst as well. And then gene expression patterns are all over the map. I don't have a whole lot of time to go into it today, but suffice it to say that the gene expression patterns from the plants with the fungi are as different between isolates that we put in the fungi as they are between non-symbiotic and symbiotic plants. They're completely different, and the gene expression patterns differ across the metabolic spectrum as well. On the metabolic side, same thing happens with the gene expression, but generally what we see is an increase in plant growth rate, increase in biomass, a decrease in water consumption. The seedlings that are symbiotic can consume as little as 50% less, or as much as 50% less water than the non-symbiotic plants. No changes in photosynthetic rates or metabolic efficiencies, but there's a dramatic increase in nutrient use efficiency, and I'm going to come back to that at the end of this talk. Okay, so based on all this research that we and others had done in 2008, we designed a new strategy to look for endophytes that would be applicable in agriculture. We felt as though we had kind of come across something that had the potential to mitigate impacts of climate change. And so we designed a strategy based around 30 criteria that we use to identify appropriate endophytes that would stay in the plants throughout the growing season, that would confer multiple stress tolerances, and that could be commercialized as well. And so we started field testing in 2012. We commercialized our first product called BioEnsure in 2017. Just a synopsis, of the acreage that we've went on in 2017, 19, or 18, and what we're going on to this year as well. The traction for this type of product is growing pretty dramatically, unfortunately, because there are places around the world now that are being terraformed by all this greenhouse gas material and transitioning from productive agricultural arenas to marginal arenas. Okay. Well, BioEnsure is comprised of a very unique set of fungal species of Trichoderma. They are all endophytic. They appear to have no ability to act as biopesticidal agents. They have no ability to grow in soil. They are devoted to living in the interstitial spaces or the dead spaces, the non-vascular apoplast of the plant. There's some other things in here as well, but those are kind of the key components into them. They pass the 30 criteria, and we've been developing them ever since. So I want to take you through what we have done and what we've seen, going from very basic research to a very applied arena now, where we're now selling a product that consists of a complex of fungal endophytes that can either be put onto seeds or mixed with fertilizer and dribbled in furrow on seeds when farmers are planting, or sprayed directly on plants. But what we've seen out there is we see really nice abiotic stress tolerance against temperature, drought, salinity. We see really nice nutrient use efficiency as well. So let me take you through some of this, the data. These graphs are all the same. What each bar shows you is a % yield difference between treated versus untreated plants or seeds. And then there's some other information there as well. It tells you about the crop. The win rates are up there as well. The win rate is an arbitrarily defined rate that we set of all these trials that are out there. The win rate is anything above 2% yield increase or 5% yield increase, whatever that is set. But most importantly, I want to show you in a contrasting way as much as possible what happens in a monocot and a eudicot treated with the same material. And this is a set of data where we've been able to quantify the type of stress and the extent of stress on the plants during the growing season. And as you can see, what happens is as stress goes up, so too do the yield benefits. And this is seen in both of these plants. In the yield, the average yield increases are fairly significant. I'll show you some more significant increases in a minute. But this is typically what we see. As stress goes up, benefit goes up. When stress goes down, guess what? Benefit goes down as well. So we drop from high, I'm sorry, let me just skip this one and we'll go on. This is just a diagram kind of showing that. And as stress goes down, so too do the benefits go down as well. So we drop from these high, or we go from these high yield increases to, when stress is low, an average of maybe 3% yield increase. We also go from win rates of above 90% to around 70%. So it's not to be wholly unexpected, but this is what we've been seeing. I'm going to come back to this low yield increase here at the end. But this is what we've seen in a wide array of plants that we've studied and done field tests on in various locations around the planet. We've tested about 25 different species at this point. They all seem to behave similarly. Okay, so I said that, as yield goes up, or as stress goes up, yields go up as well. In the U.S., it's been a little bit challenging until recently to find a lot of areas that are high stress to do testing. And so we were able to procure a grant from USAID called Securing Water for Food to accelerate the development of BioEnsure to take to poor rural farmers. And 1 year in the grant, they kind of changed the paradigm on us and said we had to go do it in India. It wasn't part of the grant, but we were able to go there and treat seeds for farmers in these small rural communities. These are farmers that maybe have 2 to 5 acres of land. They're very poor. They can't afford to buy new market seed, so they carry over seed from previous seasons, and those are all the reasons that they grow these seeds. And so in this area we're working, they had 2 staple crops. One is pearl millet, a monocot, one was mung bean, a eudicot. And we treated, I think, 300 farms is what we went on to in total. You can see the stress there was pretty impressive. By the time you get up to this part of India, the monsoon peters out and rain becomes very intermittent. Temperatures are typically in the 40s, at least when we're there working, they're always in the 40s. So it's hot and dry. And what that translates to, we didn't realize it till later, was that, you know, there have been some fantastic studies with rice showing that nighttime temperatures are more important than daytime temperatures for impacting the rice crop. So at night, for every degree above 24°C, I think, there's a risk of losing 10% of your crop, maybe 5 to 10%. And so this is where we learned that the high temperature protection was 24/7. Because we've done this with rice in India as well, and we seem to completely eliminate that great impact on fertilization that occurs with high nighttime temperatures. But the other thing that we learned here, we're going in, I won't tell you all the things we learned, but one of the things we learned, because we went in with the perfect Western scientific perspective, we told the farmers exactly what we wanted them to do, plant treated seeds next to untreated seeds, and just give us the data. That's what we're looking for. And 20% of them did that without hesitation. But the other 80% had bigger things to test. And one of the major things they had to test was, what happens if I treat my carryover seed and compare it to untreated fresh market seed? So that's about an 8-fold difference in price. And what they found was that the treated carryover seed performed at least as well as the untreated market seed. And so they were pretty ecstatic about that. The other thing they found was that because fertilizer is always in short supply there, they wanted to see if they put treated material in either in low fertility soil or just didn't fertilize it with manure or urea that they use, what would happen? And this is the only graph that I've ever put up where I'm super excited about the zeros. Because whether there was a benefit or not, these are 10 different studies that we did there with these guys, or that they did, where there was no fertilizer or low fertility, and the treated material performed at least as well as the untreated material that went into fertilized ground. So the nutrient use efficiency that the fungi bring to these plants seems to be able to mitigate the poor fertility, if you will, or a lack of fertilizer. And this then became stimulus for us to start thinking kind of way beyond what we started with. Now, we feel as though we've built this fantastic platform for abiotic stress tolerance. But to go through the rest of this century and deal with the things that are changing and deal with the environmental concerns and issues that have come about over the last 100 years in agriculture, we started looking at using this nutrient use efficiency to build synergistic microbial consortia to blend with these fungal endophytes to see if we could begin to replace synthetic fertilizers altogether. So this is just an example, a little bit of data here, showing the type of studies we're doing. So they're, the red bars are the controls or the checks. BT stands for Biotango, our bacterial consortium, as we're calling it. BE is the BioEnsure, and then BC is a combination of both Biotango and BioEnsure. And basically what we're seeing in various crops that have been studied now is that 1 + 1 = 3 or 3 to 5 is what we're seeing. So we're seeing a synergistic boost in the yield. And again, these are all yields and yield differences between them shown in the numbers up above. So you can add bacterial consortia to this small fungal consortium and achieve, I guess, and address, if you will, ecological functionalities that the plant requires or the plant has. And through that, I think we can make some really significant changes in how we produce food and the environmental impacts of agriculture. I want to stress, I don't think I have to tell any of you guys this, but this is not a solution to climate change. This is a mitigation of climate impacts. And we know that we can use this for some period of time in the future, but it will not abate the need to diminish our flow of greenhouse gases into the atmosphere, irrespective of what our president says. And with that, I want to thank you. There's a number of people involved in doing all this work. There are 20 of us in our company now, and we're located in Seattle, mostly scientists with a couple of exceptions. And I'd be happy to answer any questions if you have any. Thank you.
Thank you, Richard. Other questions? You are.
Great talk, Rusty. Do you hear me? Yes. So you started with Corvularia, right, and ended up with a collection of Trichodermas, and you focused on physicochemical characteristics of environment. But Trichodermas are microparasites. Majority of them are microparasites. Have you explored impact of these strains on resistance to pests, various kind of pests?
You know, we have. We've done all the, the lab studies looking at interactions between the strains we use, other trichodermas, other pathogens, other fungi. We see no negative impact of the fungi we're using against other organisms. We do see negative impacts of other organisms against these strains. They don't appear to have any microparasitic capabilities that we've been able to visualize. In the field, we haven't seen any protection against any diseases that have occurred out there. There may be a delay sometimes in the onset of some foliar stuff, but we think it's just because there's more chlorophyll in the plants and the plants are healthier and they're less stressed. But they catch up pretty quick.
So once you apply BioEnsure in the field, will the Trichoderma mix stay for a little bit until the next season? Or also what happens with like either vertical transmission within a crop, or if you have a crop that can be propagated clonally, whether this will stay?
Good questions. So what I didn't mention was that these strains, they reside in the non-vascular apoplast. I think I told you that. They cannot get past the floral abscission layer, so they don't go into fruit or seed. They can't get past, I don't even know what it is, but they don't go into tuber or into root crop, and they don't get past the Casparian strip into the vasculature. So they are not vertically transmitted. It's a good business model, but we didn't design it that way. Something else did. But in perennial plants, then we have a series of long-term studies going now to address how long they stay in there. But it appears, based on the benefits that we're seeing, that like if you go into something like alfalfa, it'll be there until they replant, or into pasture grass as well.
Are you concerned about introducing species elsewhere?
I'm sorry?
Are you concerned about introducing species elsewhere?
I'm always concerned about introducing species elsewhere. So that's one of the reasons why we know these are not soil competitive, is because we've done a number of studies looking exactly at that. If we put seeds in the ground, how long can we pick those strains up out of the soil? And they disappear really quickly. So we have markers that we can measure them with. Is there any risk? There's always risk, but I think the risk is very low, and it doesn't keep me awake at night like it did initially. So we're very comfortable on that front.
One more question. Two more questions.
So I may have missed something, but I thought you just said that it wasn't actually transmitted to the seed.
That's correct.
But yet when they did the experiment where they subsequently planted the seeds, right, in India, they actually saw an impact.
Okay.
So, yeah.
Sorry, I wasn't very clear about that. So what they do, the farmers there, they grow their crop and they retain some of it to plant in the next growing season. But the seeds they retain do not have any of the organisms in it. They just don't get, they can't get past the floral abscission layer to get into the seed. Does that answer?
So to be clear, when they planted, I thought you said they planted the next generation of seed that they harvested.
No, they planted the next generation they harvested, but we treated that generation before it went in the ground. Yeah. So the treatment has to be applied every year to annuals.
One more. White shirt.
Yeah, these seem to be really successful symbiosis. I was wondering, why don't these exist already? Why did you have to create them?
Well, you mean, why do we have to create them in agriculture at all?
Where did the, maybe I missed it, where the fungal associates come from? And
All of the fungi that we work with came from native plants. And we looked at across a wide array of habitats. So what we've contrived is that we put them into agriculture. But in agriculture, we have done a superb job over the last 100 years because microbes are evil and bad of eliminating them, right? So we've enriched for pathogens and we've gotten rid of lots of beneficials. We're putting them back now. That's really what it comes down to.
Thank you, Rusty.